Hi everyone, my name is Celeste Parra. I just finished my first year here at UCSB in the biology program, and my project this summer involved investigating the functional roles of delta-catenin. I worked in the molecular, cellular, developmental biology program under Elmer Guzman in the Cossack lab. So there's one thing that schizophrenia, autism, and Alzheimer's disease all have in common, and those are just simple mutations in the brain. Neurodevelopmental and neurodegenerative diseases like these have been around for a long time, but it has only been in the last 50 years where scientists have done extensive research into these diseases and how they come to be. And all in this is for us to know more about the brain. There's still a lot of things that we don't know about it, and a lot of interactions that we're still investigating to determine how these diseases develop. And we want to do this in order to be able to find a cure for these uncurable and debilitating diseases. So my part in this is to investigate how delta-catenin, a synaptic protein that's found to be a key player in the regulation of dendrites, synaptic density, and overall function of the neuron, how this specific protein, delta-catenin, plays a role in the development of these diseases. So delta-catenin, there has been a lot of work done with this protein in mouse cells and in regards to knocking out the protein and seeing what effects, what downstream consequences the absence of this protein causes. And what we want to do is the same thing, but in human neurons. There has been little work done in that field. So mutations cause a lot of problems. For example, mutations in the gene of delta-catenin itself can cause too few or too many synapses, and this is directly linked to autism. While a mutation in a gene that interacts regularly with delta-catenin causes plaques and tangles in the brain, and this is directly linked to Alzheimer's disease. By no means delta-catenin is the major player in the development of neurodegenerative diseases, but it's important for us to be able to understand how all of this how delta-catenin interacts with its surroundings. So the bigger project here is to understand how the downstream consequences of when delta-catenin is knocked down in stem cell-derived human neurons, and located on chromosome 5. The way that this would work is on the specific gene, a complex would bind to the gene, which would then inhibit the transcription of the gene. So knocking down means exactly this, inhibiting the transcription. And then this would stop the gene from being transcripted, translated, and then produced into a protein. So the protein would no longer be present. And what we want to do is find a specific sequence within the gene that transcribes for delta-catenin the best. And would then, whichever sequence transcribes for it the best would be repressed the most effectively and stop the protein from being produced. So that is my project for this summer. It's just basically confirming the knockdown of the best knockdown using a different locating protein locating guide RNA to see which one is the most effective in repressing. So my methods are very lengthy, so I'll give you an overview of the most important ones. So first, in terms of getting the neurons ready, we chose to use iPS cells, or induced pluripotent stem cells, which are basically a type of cell that can be turned into many different types of cells. They have a good predictive capacity and are good for studying human diseases in vitro. So we turn those into neurons. Going into more of the repression process, we used a complex formed of 3 different things. We used a CRUT repressor that it does what the name says, it represses, and the guide RNA was used to locate that specific sequence of the 3 sequences that we chose. And then this is called a CRISPR-I. You may have heard of the CRISPR-Cas9 system, which is a relatively new technology used for gene editing. And the way CRISPR works is that it binds to a specific target in a gene, and it cleaves it at a certain point. Well, CRISPR-I uses the dead Cas9 system, which binds to a specific area of the gene, but it does not cut it. So we used these three together, and then we used our 3 different guide RNAs that coded for the 3 specific sequences. And so the guide RNA was used to look at the gene, and then the Kreber pester was fused to the dead Cas9 system, and that was the repression machinery, which was used to stop the protein from being produced. So the second step is we wanted to measure the relative expression between our experimental sample, which had the delta-catenin protein knocked down, and the control, which did not have anything knocked down. So we did this by amplifying the amount of delta-catenin mRNA. And the mRNA was used as a tool to see the relative expression. We didn't measure the relative expression of the protein itself, but the mRNA has a high correlation with how much protein there is. So the way, and we use qPCR, quantitative polymerase chain reaction, for this, and the way that this worked is that the delta-catenin DNA was denatured, meaning it went from being double-stranded to single-stranded, and then a primer that targeted a specific area of DNA annealed to the DNA and then made it replicate many times over. So going into, backtracking a little bit, these are just images of how the stem cell differentiation into human neurons works, and how the morphology changed from when the enzyme was added to begin the processes. So you can see the cells starting to spread out, and by day 7, they look much more complex and much more like neurons. So these were the ones that we used in our final experiment. But before testing how delta-catenin, the absence of delta-catenin affected these, we wanted to use different cells that are easier to work with. And so we used H4 cells, which are just different cells found in the brain. So we wanted to confirm that the guide RNA was being expressed within the cell. So in order to do this, to get the guide RNA into the cell, we created a plasmid, which is a circular piece of DNA. And this plasmid had to be taken up by the cell. And in order to see if it was being expressed, we added a code that coded for blue fluorescent protein and as well as their guide RNA. So if they fluoresced, we knew that these both were being expressed. And they did on day 2. So looking at our qPCR results of the amplification of delta-catenin mRNA, we can see that in H4 cells, we can see that our results are kind of inconclusive. You would expect that the control, which did not have anything targeted, there was no amplification of mRNA, would be higher than the ones that targeted the delta-catenin. And guide 1 actually replicated many times over, and these 2 also did not show any significant differences with compared to the control. So this test was a little bit unreliable. So we chose to, and because of this, and because delta-catenin is just much more highly expressed in neurons than it is in H4 cells, we decided to move forward and test all 3 guide RNAs again in neurons. So the results for this actually showed positive, there were positive results in that the control was, the 3 different guide samples showed a relative